All right, so let me draw out what would happen to these chromosomes. So we have a big and a small chromosome, 4 total chromosomes in a diploid organism, so 2 pairs of chromosomes. And the 4 genes that are shown here are all heterozygous. So we could track them all in the process of mitosis. Specifically, as I mentioned, I'd like to cover just metaphase. So let me get up our drawing pad here, and we'll see if we can do this. A big one. Just start by writing our genotypes here. So we had a little q and a big T, big chromosome, and a big Q and a big T, big chromosome, and little chromosome. 2 versions were little r with big N and big R with little n. So that was them in their unreplicated form. So I guess an important thing to be able to do is to be able to write them out as replicated chromosomes. So we're going to get a big chromosome, and that one's replicated. Big chromosome, replicated. So still 2 chromosomes that are big and 2 that are small. Just each one's going to take on this more X shape. And I can just copy the alleles. So remembering that a copy of little q should also be little q, a copy of little t, also little t. Okay, so this is them. I just wanted to show them replicated for starters. And now we can go into a cell and show you how they might line up in a cell. So let me draw the boundaries of a cell. I got awful big. Okay, so I can draw a dotted line for the midpoint of this cell. And it doesn't matter what order we put these in. So I can put big, little, big, little. And sometimes I like to change it up just to highlight that the cell is not imposing any order at this level. So each chromosome is really doing its own thing. And the important part is that the 2 identical halves of these chromosomes end up going to their own cells. And that every cell that results from this gets 1 full set of every gene. So one way to think about this, we could look at the genotype of the starting individual. And as I mentioned, we're heterozygous for everything. So there's a big Q and a little q, there's a big T and a little t. There's a big R and a little r, big N and a little n. Right, so this is, that would be the genotype of this individual cell that started. And the end result after metaphase comes anaphase, and all these chromosome bits are going to split apart and go to opposite ends of the cell where they're going to end up in their own cells. So those ending cells are going to get a big Q, check, a little q, check, a big T, check, and a little t, check, and a big R, check, and a little r, check, and a big N, and a little n. Okay, so all of those are present there. Here we go, big Q, little q, big T, little t, big R, big N, little n. So all those same alleles are showing up in this resulting cell. So after we split up along the midpoint, distribute these chromosomes into the resulting cells, those resulting 2 cells are going to have the same genotype as the starting cell. So that's how you would draw a cell with these alleles going through metaphase.